One of the reasons antimicrobial resistance has become such a huge issue is because of how widely it spreads in a short amount of time. This is especially prevalent in areas with high concentrations of harmful microbes and antibiotics. Can you think of a place where a lot of pathogens and antibiotics coexist around humans?
Well, I think hospitals are a prime example due to many people who are being treated for infections daily. It means there are also a lot of bacteria in constant interaction with antibiotics that generate a selective environment, which leads to acquired resistance. This can create large consequences for us. Like in 2019 alone, antibiotic resistant pathogens were found directly attributed to approximately 1.3 million deaths. Under the appropriate selective pressure in a favorable environment, it is not a difficult task for bacteria to exploit these conditions and share antibiotic resistance genes with each other in a process called horizontal gene transfer, which allows them to counteract antibiotics and rapidly evolve within these clinical environments.
Uh-oh,
What?
low battery. If we want to continue playing this game, we should go home and continue on the console.
Let's hurry so we don't lose our progress.
Olivia, I'm gonna transfer all this data off so we can continue playing in the console.
Good, thank you. Thanks. Just as we are physically transferring our data from one device to another, with horizontal gene transfer, bacteria are able to share genetic material to other bacteria nearby. This allows them to acquire resistance and level up to fight off antibiotics. Oh, it looks like our data transfer is done. Let's hop back into the game and see some examples.
There are actually three types of horizontal gene transfer: conjugation, transformation, and transduction. Conjugation is the most studied type, and likely the most common way bacteria acquire resistance. The best way to think of this is as if you were to share your upgrades with other nearby players or characters. During conjugation, bacterial cells make contact through their pili and transfer short segments of DNA or circular segments called plasmids of DNA.
The next type is transformation, which is where bacteria pick up pieces of DNA that exist outside of cells in the environment. This can be problematic because even if a bacterial cell dies, it can break down and its DNA is released in the environment, ready to be taken up by nearby bacteria.
Finally, transduction requires specialized viruses, called bacteriophages, that infect bacteria so they can inject DNA into their victim. Sometimes, their DNA contains antibiotic-resistant genes picked up from other bacteria they have infected. If the bacteria are more resistant to antibiotics, this also helps the bacteriophages, since they need bacteria to survive. To illustrate how horizontal gene transfer drives the spread of antibiotic resistance in pathogens, let's discuss the case of Staphylococcus aureus, a very common human pathogen that can occasionally cause skin infections, respiratory infections, and food poisoning-related sickness.
In 1959, doctors started using the antibiotic methicillin to treat infections caused by penicillin-resistant strains of Staphylococcus aureus. However, genes that give out resistance to this antibiotic were horizontally transferred into a human-associated Staphylococcus aureus strain a few years later after methicillin introduction. This resulted in the well-known methicillin-resistant Staphylococcus aureus, or MRSA, which is a serious health threat and a common inhabitant of hospital environments that causes hundreds of thousands of infections annually. The antibiotic vancomycin is the last resort antibiotic to treat MRSA infections, meaning if this doesn't work, well, game over.
However, vancomycin-resistant Staphylococcus aureus, or VRSA, evolved from MRSA through horizontal gene transfer of a plasmid from a fecal-related bacteria called Enterococcus fecalis. VRSA became a superbug that displays resistance to several antibiotics. These bacteria are also called multi-drug resistant pathogens. The more drugs that a pathogen is resistant to, the less chance we have of treating infections. This is an example of how bacteria use horizontal gene transfer to select and acquire beneficial genes in order to survive and spread in a particular environment, which makes it a lot more difficult, and sometimes impossible, for humans to fight off infections. Even good bacteria that are exposed to antibiotics by human and animal consumption can experience selective pressure, potentially spreading resistance throughout our microbiomes.
Probiotics, such as lactobacillus and bifidobacterium, are common inhabitants of the human intestinal microbiome and can be administered in adequate amounts to provide health benefits for the host. Their intrinsic resistance is not a huge concern on its own, but if they contain mobile genetic elements with resistance genes that combat commonly used antibiotics, these generally helpful bacteria do not differentiate between good and bad bacteria meaning that they may pass on that resistance to bad actors, like food or gut pathogens. When those newly resistant bacteria exit the body through fecal waste, they eventually travel to wastewater treatment plants. On top of that, antibiotics aren't always broken down in the body, so drugs that stay intact and their associated leftovers, or residues, are also excreted from the body. These wastewater receiving treatment plants are kind of like a hotspot for antibiotic resistance.
The presence of antibiotics and their residues leads to selective pressure across massive populations of bacteria that are commonly found in human waste. Already resistant bacteria will dominate and pass down these abilities to their daughter cells and share with nearby bacteria through horizontal gene transfer. In an environment like a wastewater treatment plant where you will find a higher amount of bacteria, horizontal gene transfer occurs at much faster rates. Wastewater treatment doesn't eliminate all resistant bacteria, and they are also not designed to efficiently remove antibiotic-resistant genes, so some of those bacteria and genes make their way into the environment where resistance is spread to naturally occurring microbial populations. Wastewater treatment plants and their sewage are considered important reservoirs and sources for the spread of antimicrobial resistance in the environment.
To sum it up, where you have high use of antibiotics, intrinsic or acquired resistance, and bacterial populations, you're also inevitably going to create the perfect recipe for extensive antibiotic resistance expansion in the environment. So, what does this mean for us?